Good morning, good afternoon, good evening to all that are joining from all across the world. My name is Florent Ginou, and today it's my pleasure and honor to host and introduce our global IMMUNOTALK speaker today, Professor Jony Kipnis, or Jony Kipnis for the friends. So before doing that, a reminder that we will have Professor Bruno Silva Santos next week on January 26th. So please stay tuned and join us so next week on January 26th. So today we have Professor Jony Kipnis from Washington University presenting live today from St. Louis. Dr. Jony Kipnis is a BGC investigator, Alan and Edith L. Wolf Distinguished Professor of Pathology and Immunology and Professor of Neurology, Neuroscience, and Neurosurgery at Washington University in St. Louis School of Medicine. He is also like an inaugural director of a Center for Brain Immunology and Glia, big at Washington University. So Jony is born from Georgia and also lived in Israel, graduated from the Weizmann Institute of Science in Israel, where he was a Sir Charles Clore Scholar and a recipient of Distinguished Prize for Scientific Achievements awarded by the Israeli Parliament, the Knesset. So Kipnis Lab focus on the complex interaction between the immune system and the central nervous system. The goal is to elucidate the cellular and molecular mechanism underlying these interactions in neurodegenerative, neurodevelopmental, and mental disorders, as well as in physiology, including healthy aging. So his lab showed that the brain function is dependent in part on the function and integrity of the immune system, and that immune molecules, such as cytokine, can play a neuromodulatory roles. So the fascination with immunity and its role in neurophysiology is what brought the lab to a breakthrough discovery of meningeal lymphatic vessels that hopefully, like Jony will speak today, that drain the CNS into the peripheral lymph node and serve as a physical connection between the brain and the immune system. So these findings challenge the prevailing mechanism underlying CNS immune privilege and open new avenues to mechanistically study the nature of the neuroimmune interaction under physiological and pathological conditions. So the implications of this work are broad and range from autism to Alzheimer's disease through neuroinflammatory conditions such as multiple sclerosis. And recently, the Kipnis lab has also identified the skull and vertebral bone marrow niches as local immune reservoir for the brain and the spinal cord, whose role in neurological disorder is yet unknown. So among other awards, Dr. Kipnis is a recipient NIH Director Pioneer Award for 2018 to explore in more depth neuroimmune interaction in healthy and diseased brain, and a National Institute on Aging Merit Award to study the role of meningeal immunity and lymphatics in aging and AD. So again, Jony, thanks so much for accepting our invitation and for giving a global IMMUNOTALK that will be entitled Immunological Uniqueness of Brain Borders. But just before we start, we have a tradition to ask a question for you, and this question will be about your career, and it will be, could you share one of the most impactful decisions in your scientific career? Jony, please.
Thank you very much, Laurent, and thank you for the organizers for inviting me. So it's really a pleasure to be here. And for your question, I have two answers. The first thing was, I guess, when I started my lab and I was told that I should look what NIH funding announcements are and tailor my research towards that. And it didn't really make any sense to me. And I decided to stick to what I thought was interesting, and somehow I managed to survive. And then maybe the second thing is when in 2012, I was at the conference and there was a group of young immunologists standing there, and I approached them and I said, I want to introduce myself. Actually, it was you and Burkhard and a few others. And so we started to talk, and you told me, oh, I thought you were this old guy. So nice to meet you. So I think that's where the friendship started. So that was another good impactful step in my career. So
my apologies again for this. Anyway, thank you for sharing with us, and please start to share your slides, and the floor is yours, Jony, please.
Okay, thank you, Florent. And I guess you can now see my slides, and I'll let them play. Well, again, thank you so much for the invitation, and thank you to Elena and to Carl, of course, for starting this amazing adventure of your global IMMUNOTALKs, and to everybody else who is now really taking kind of following their steps and organizing this really exceptional seminars that I think everybody is really excited to be part of, whether on this end or the other. So I'll talk today about immunological uniqueness of brain borders, like Florent said. Maybe I'll start with my disclosure slide quickly. And let me start with a little bit of introduction. I know it's a diverse audience, so I will try to kind of present this story more conceptual, maybe less going into details. And I really hope that we can really discuss the more specifics over Twitter in the next couple of days or whatever. So central nervous system, or CNS as we refer to it, is actually immunologically privileged site. And what is immune privileged organ actually? There is probably, if you ask 10 different people, you probably will get 12 different answers. But the very first observation for the brain, actually, for the eye it happened even before, but for the brain, the probably first observation was published about a little bit over 100 years ago in 1921, when a Japanese scientist Shirai showed that if you take sarcoma cells from rat and you put them in a brain of a mouse, actually it will not be rejected from there as efficiently as it would be rejected from the periphery. And that kind of was the beginning of thinking that there is something unique about immune response in the brain. And 2 years later, there was a group of Americans, Murphy and Sturm, who followed up on these observations and found something very similar. And then 20 years later, when Peter Medawar, or Sir Peter Medawar, was doing his experiments, of course, Peter Medawar got Nobel Prize for understanding mechanism of peripheral tolerance. So he was working on transplantation of skin. And so if we translate it maybe to current times to make it a little bit easier, if you take, for example, a piece of skin from a white mouse and you put it on a black mouse, of course, the rejection will be very, very fast within very few days. But if you put the same piece of white skin into the brain of the black mouse, it will stay there for a very long time, which is, again, similar to what others showed with tumors. What was different in Medawar's studies is, of course, he did another experiment when he took his skin from a white mouse and put it in the skin of the black mouse, on the skin of the black mouse, and in the brain of the black mouse. And now, guess what happened? Well, both sides were rejected. So, of course, today we can say, okay, so if you introduce something foreign into the brain, it probably is not very efficient in eliciting the immune response, priming the immune response. But if you prime cells in the periphery, and then there is a pathogen or there is an antigen in the brain, then the cells will be able to go and migrate and maybe reside in the brain. But of course, back then, in 40s, it was way above the knowledge of immunology for back then. And so this last part of Medawar's work really became almost ignored for years. So this idea of CNS immune privilege kind of pertained very strongly in people's minds. So CNS is this unique organ. If there are interactions between the immune and the immune system and the brain system, that usually happens during maybe like MS or some other pathologies. So this idea was that the two systems always interact in pathology and cause pathology. Of course, there is a very unique structure in the brain of the vasculature. It's called blood-brain barrier. It's actually a very unique physical barrier that is formed at the level primarily of blood endothelial cells, which have unique tight junctions in the brain. And they supposedly preventing immune cell entry into the parenchyma of the healthy brain. But we know that this is probably not the case, because again, if you activate the cell in the periphery, the cell will be able to cross the parenchyma. Now, still in the brain, a healthy normal brain, at least in the mice, we do not see cells, resident lymphocytes in the brain, right? And we will talk about this in a second. And the other reasoning for why CNS could be this immunologically unique, let's call it immune privilege, is because CNS has no lymphatic vessels and therefore no direct physical connection with the lymph nodes and the immune system. And this is also not really correct, because even though there are no lymphatic vessels within the brain tissue, there are lymphatic vessels outside of the brain tissue that actually doing the drainage. And I'll talk about this a little bit in my talk. So if we can now look at the brain a little bit in a cartoony way, we have the brain tissue, the parenchyma of the brain, where the major immune cell are the microglia. They're not the only one, because we also have perivascular macrophages, but those, of course, are not really part of the parenchyma. They are in this perivascular space. But there are also reports of other cells in the brain. And so in aged brain, and definitely in aged human brains, you can find many more cells. You can find T cells and NK cells and gamma delta T cells. But again, these cells will be there in a very, very small numbers. The majority of the cells in the parenchyma, about 10% of the brain cells, are the microglia, the tissue resident macrophage. Now, what happens is, however, is that if you just look outside of the brain tissue, so the brain parenchyma is here, the skull is here, and between the two, there is a layer. It's a three membrane kind of layer tissue called the meninges collectively. So we have the dura mater, the arachnoid, and the pia mater. And of course, the just above the brain flows cerebrospinal fluid. So here is the pia attached to the brain. Then we have this fluid, cerebrospinal fluid, then the arachnoid, and then the dura. And so we always thought that this cerebrospinal fluid, provides buoyancy to the brain, maybe remove some toxin, etc. But really, the understanding of the fluid understanding of the kind of new understanding of what this fluid could do came later, and we'll discuss this a little bit as well. Now, what is interesting is if you look now into this area of meninges, in a normal healthy young mouse or even a healthy human, what you will see is very nice variety of immune cells, all kinds of cells you will find there, CD4s and CD8s and gamma deltas, and and different types of monocytes and all kinds of DCs and B cells, and really the diversity is so diverse, as if you're looking into some a barrier tissue like a gut or a lung. And the question is, what are the cells doing there? And so unfortunately, I will not be speaking about the function of the cells today, but we are very interested in different populations of these immune cells, their cytokines that are produced, and how the cytokines affect the brain. So we showed some produced by CD4, some produced by gamma delta, and many others. So today I would like to talk to you a little bit more about this understanding of really whether I'll hope I'll convince you that these cells in the periphery of the brain are really put there to provide immune surveillance to the tissue. So as you know, every tissue has two types of vasculature. We have the blood vessels and we have the lymphatic vessels. And the blood vessels, of course, bringing all the goodies into the tissue, nutrients, oxygen, glucose, and of course, immune cells are coming into the tissue with the blood vessels. Now, most of the things are coming out of the tissue. Majority of the interstitial fluid, which is kind of carrying metabolites and all the waste products from the tissue, most of them are coming out of the tissue with the lymphatic vessels. And every tissue really is highly vascularized by both vessels. Just think of your household. You have water pipes and sewage pipes, right? So water comes in, and then all the sewage is leaving the household with your sewage pipes. Now, brain does not have lymphatic vessels. So the question is, okay, so is brain not being perfused? Well, that's kind of hard to believe, right? Because brain is a very active organ. So how is the brain being perfused? And how is it could this process could be efficient without having lymphatic vessels in the brain parenchyma? By the way, I must say that there was one paper recently that claimed for lymphatic vessels in the brain parenchyma, but they just finally they used LIVE1 as a marker, which is a marker for perivascular macrophages. So really, up to today, there is no report of lymphatic vessels within the brain parenchyma. Yet the brain is a highly active organ, produces lots of metabolites, and the question is, how is brain being perfused? So a couple of years ago, Mike and Nidegard came up with this really beautiful hypothesis, and actually she showed some very interesting findings. And what she claims is, and I think many labs now, including our own, reproduce these findings, is that the brain, I told you, is kind of swimming in the perivascular, in a CSF, in cerebrospinal fluid. And so the CSF actually can go and wash the brain through. And the way the CSF will get into the brain to wash it through, it will come with arteries. So what arteries are coming through leptomeninges, and then they're diving into the brain. As they dive into the brain, there is a space between the arterial cell wall and the brain tissue, and it's formed by astrocytes, which are the glia cells. So you have this kind of space between the vasculature and the actual tissue, which is filled with this new CSF, which is being produced. Now, because arteries are pulsating, the CSF will be kind of pushed out and will end up in the parenchyma of the brain, and it will be pushed in, so it will create a little bit of a push, will diffuse to the tissue, will collect metabolites, and will be recollected at the perivascular space. Arguably not a very efficient perfusion, but remember that lymphatic drainage is also all based on diffusion, right? Because there is no lymphatic pump that pumps the metabolites out. So it's all based on this more passive flow of the metabolites. And that's what's happening most probably in the brain. And Mike can show that aquaporin expression, which is the water channel expressed on astrocytes, is very important in mediating this effect. So this is what the system is about, and the system is called lymphatic. G stands for glia because astrocytes are forming this part of the space, and the lymphatic is because it serves the function of lymphatic. But then If the clean CSF comes in, washes into the brain, and then comes out as dirty CSF, where does the dirty CSF go? And there are different proposals. It could go through this route and that route. And unfortunately, we couldn't really establish any of these routes in our lab, in our hands. And so back then, Antoine Louveau was a postdoc in the lab. Today, he is a PI in Cleveland Clinic. And he was very interested, like I told you, in this covering of the brain and the meninges. So when he learned it, so what he learned to whole mount the whole covering of the brain as a whole mount of the tissue. So you can see these vessels here. This is labeled for CD31, so all the vasculature is labeled. And you can see that these vessels, the sinuses in the middle of the covering of the meninges, are the major venous structures that remove all the venous blood from the brain, and then it goes to jugular and then out. So everything from the brain will come through those sinuses. What Antoine notices that, first of all, the sinus, so this is a perfused mouse. Now we're looking into the sinus in a zoom in, and you can see there's a lots of immune cells along the sinus, and they are all abluminal. But there are some cells which were next to the sinus within the vessels, and so they were not perfused. And we said, what is it not perfused? How come vessel not being perfused? Because we know there is no lymphatic vessels in that area of the brain. Well, at least we knew so, but we labeled for LIVE1, which is one of the markers. And indeed, we show the structure, we show the network of something that looked like vessels. Now, remember, LIVE1, like I told you, is also a marker for macrophage. Many perivascular macrophage were labeled with LIVE1. So, but you know, this looks like of course, this looks more like a vascular structure, but we also labeled with all other markers for lymphatic endothelial cells. So there is no real one single marker for lymphatic endothelial cell. You need to label with 3, 4, 5 markers. And then if you have co-labeling, only then you can claim, yes, this is really a lymphatic endothelial cell. So they express PROX1, they express protoplanin, tissue lymphatics all do, and they express VEGFR3. And actually, if you give them VEGFC, they even expand, and so they respond to molecule. They also express CCL19 and CCL21, which are the molecules that can recruit cells into lymphatic vasculature. And so, long story short, now we have lymphatic vessels outside the brain tissue, but are they draining the CSF? So to answer this question, we inject green dye intravenously. So now all the blood vessels are green, and we inject red dye into the CSF. And now what you see is that there is a flow of the tracer in the blood vessel, and there is a flow of the red tracer from CSF within this beautiful lymphatic vessel. So the vessel is functioning. So these findings were initially questioned, but then finally very quickly reproduced by Cariel Italo group, who is, of course, a very big name in lymphatic biology. He published very soon after our paper, and so showing basically very similar findings. Then these vessels were also found in zebrafish. Actually, there were 3 papers from 3 parts of the world. There was Europe, Australia, and US, all showing that really these lymphatic vessels also present in, actually I was confused, Ben is Australia, previous one was US, that all these vessels are present in zebrafish, so they're evolutionarily conserved. The system also exists in rats, and now we're showing in squirrels and whatnot. So it's, but then most importantly, maybe that Danny Reich and now many others have shown the structure also present in non-human primates, but also in humans. And what was really striking, and even in humans, as you can see here, it's very similar to what we see in mice, and most of the meningeal lymphatic network really goes along the sinuses. So what is so unique about the sinuses? And even before then, how does CSF really get access into this meningeal lymphatics? Because what I told you is that the vessels are along the sinus, and that's what we see in humans. And sinus is in the dura, and there is between the dura, underneath dura, there is arachnoid, which prevents CSF to get into the sinus, into the dura. So what's happening there? So if you're reading a textbook and you're reading old papers, what they will tell you is that this middle structure, the arachnoid, forms those unique granulations which will shoot CSF directly into the blood vessel, okay, through those little holes. So something like that, basically. So here is our lymphatic vessels are next to sinus. This is our big sinus, okay? We're kind of looking a little bit, it's a bit weird angle, but it's kind of just to orient you. So basically then we have this unique arachnoid granulation, which is sticking all the way into the sinus. Now, and then what some people thought is, well, maybe when we put tracers, they just come into the sinus because that's where CSF goes, and then somehow maybe leak out into lymphatics. But regardless, we see them coming into the lymphatic vessels. So I said, well, if anything, we don't care really how much gets into lymphatic, as long as lymphatic are sampling the antigens, they will be able to present them to the immune system. But then there was a paper that came out from Steve Prook's lab, and he actually showed that all CSF, he claimed, that all CSF goes into lymphatics, which also will support what's happening in other tissues, because the majority of interstitial fluid goes into lymphatics and not into the vasculature. Now, to be honest, this structure always bothered me. Number one, what bothered me the most is how come you make a major blood vessel and then you just poke it with those protrusions of arachnoid? How do you seal this area? How do you seal that the blood doesn't come out? That never made sense to me. Well, because I didn't read anatomy books as I should have. And I'll tell you in a second why. The other thing that bothered me is that in mice, for example, or even in baby humans, we don't see those structures. So if you don't have the structures, how does the CSF get into the sinuses? Or do we have different routes in mice and in humans? So Justin Rastenhoven, who is a postdoc in the lab, still officially, even though he's now already left to start his new lab in 11 days in New Zealand, and Tornike Mamuladze, who was our visiting scholar with us and now graduate student here at WashU. So what they did is they made this large window. They injected 2 tracers. So we're now looking from the top of the mouse, and you can see this is a sinus, superior sagittal sinus and transverse sinus, as you can see here. So we're injecting with 2 types of tracers. One is a 0.1 micrometer bead, and the other one is a very, very tiny 3 kilodalton dextran. And we are watching. And so if the literature is correct, and if the predictions are correct, and that we are wrong, then the tracers will be coming from the sinus and maybe then leak into the lymphatic, into the dura. Remember, lymphatics will be nearby. If the literature is incorrect, we will see something very different. So 5 minutes later, we don't see anything. At around 7 minutes, what we see is the tracers are not coming from the sinus. They coming kind of along the sinus in this perisinusal area, which, to be honest, made no sense, right? Because last thing you want to question are anatomy books. So, but first of all, fact number 1 is when we peel out the dura, we really see those tracers in the dura. So, which tells us, okay, from the CSF to the dura, things do go. Okay, how they cross arachnoid, we don't understand, and they clearly don't show up first in the sinus. So Steve is correct, it all may be really going into lymphatic first, and B, they do end up somehow in the dura. As we're revising this paper, which came out last year, as we were revising it, another paper comes out from a Norwegian group of Ringster and Eide, and they actually did experiments with humans. They injecting humans with tracers in the same place, intracisterna magna, so putting that in a CSF, and watching what happens with those tracers. And what they see is something really spectacular in humans. And they say here that the tracer escaped from cerebrospinal fluid into parasagittal dura, along the superior sagittal sinus at areas nearby entry of corticocerebral veins. Basically, exactly what we are seeing in our mouse studies, and we have no clue what's happening, what's going on there. So the findings demonstrate that transarachnoid molecular passage does occur and suggest that the parasagittal dura may serve as a bridging link between human brain and dural lymphatic vessels. So I also a little bit updated my anatomy knowledge, and basically there is nowhere in literature that says that granulations in humans are sticking all the way into the sinus. They come close to, but the sinus cells, but the endothelial cell wall, endothelial cells are continuous. So there's no such thing as sinus goes inside, the protrusion goes inside the sinus. It comes close to. So what we think is happening now, based on the human data and also on our mouse data and on the really anatomical structure, is that the CSF will come out through these arachnoid granulations, and then will end up in the dura, and then will be picked up by the lymphatic vessels. So we have kind of 2 stages to get into the lymphatics. In normal tissue, it would be interstitial fluid into lymphatics, and in the case of CNS, it will be interstitial fluid, which is CSF, and then into the dura, and then into lymphatics. And I'll tell you in a second why we think it's very important. So just to summarize, what we're proposing is that the clean CSF is being made in the ventricles, flows around the brain, goes inside the brain to become brain's interstitial fluid, then comes out as, if you wish, dirty CSF, and then mixes, maybe partially, maybe not at all, with the clean CSF. Which, by the way, just to think about it, when we're sampling CSF for biomarkers, which one are we sampling, the clean or the dirty? That's a question for another time. So now this dirty CSF comes out, and then it goes into the dura to become dural interstitial fluid. And only then it will end up in the lymph nodes. Now, why do we think this area of dural interstitial fluid is so important? Because what happens is along the sinus and on the sinus, like I told you, there are lots of myeloid cells along the sinus that express, as you can see here, very high levels of MHC-class II. We also see that majority of the T cell in the dura, 95% or so, are really congregating along the sinuses. Even when we induce EAE, we still, majority of the cells will be along the sinuses. And we show that around the sinuses, we have, first of all, high expression of adhesion molecules, but we also have fibroblasts, which are producing retention molecules, such as SDF1, which actually keep those cells in the area. So, and we show that actually, if you take immune deficient mouse and you give them labeled T cells, they will populate meninges primarily through, as you can see here, the tracts will primarily be along the sinuses. So what happens is when the tracer comes out through this perisinusal area, it being captured by those APCs and is being presented right there at the sinus through which T cells can very easily come. Now, remember that the sinus is really not inside the brain, it's outside the brain. So it's a peripheral blood vasculature, it's fenestrated. So if you put tracers in the blood, they will also get there. And they will also be captured by macrophages and DCs. So now you have this area at the very top of our heads that can capture the CSF and can capture the periphery and can present the both of them to patrolling cells. So this is very exciting. Like you capture the highest area in the body, you the top of the hill, which is always good if you are in a war with somebody. So you present there the cells, and then immune cells are patrolling. So we said, okay, this may be really the gateway of immune cells into the brain. And probably that's where immune surveillance is happening. And if that's true, let's take now T cells reactive to ovalbumin, which should not respond to anything in the brain. And we know from works of Alex Flugel with Harvard Beckerle many years ago, now over 20 years ago, and we know that these cells will never get into the meninges or the brain tissue. But let's now take these cells, put them in the periphery. We know they won't get, but we will inject a very, very tiny amount of actually their recombinant antigen of albumin into the CSF. And we'll ask ourselves, is this sufficient to then recruit those cells and maybe retain them there? And indeed, in control mice, you cannot find any T cells, any ovalbumin-specific T cells in the meninges. But if you give them a little bit of ovalbumin, this recruits T cells directly into meninges, and they actually acquire also residence phenotype. So suggesting that really this is the area, meninges is the area where immune surveillance of the brain is happening. And now going back to humans also, you can see here the granulations are in the humans. They, of course, don't get into the lumen because the endothelial cells are covering them quite nicely. And they're packed with MHC expressing myeloid cells, DCs, macrophages. There is also, by the way, B cells, both here and in the mouse, also expressing MHC-class II. So what we are overall suggesting is that we have those macrophages. I wrote here macrophage, it should have said APC. So when antigens are coming from the CSF, remember CSF brings everything from the brain because it washed the brain, now it comes out. So it brings now antigens from the brain into the dura, macrophage capturing it. The T cells are coming and patrolling through this area. There is interaction. And if there is interaction, then the cells stay and maybe induce local inflammatory response. Maybe they recall some other cells. They may potentially even dive into the brain from that region. If there is no interaction, they will leave. So basically, to ensure, so what we're suggesting is that immune surveillance of the brain is happening normally. But the difference is instead of making this inside the tissue, you just take out everything into the periphery, and instead of bringing cells inside, you take antigens outside. And I give this example, which people tend to like, is if you have a sleeping baby in a house and you want to host a party. Sleeping baby will be the neurons, and the party cells will be, and the party guys will be the immune cells. So instead of bringing your noisy party people into the house, you take all your treats out into the courtyard, and that's where you host your party. So again, because as we all know, immune surveillance of the tissue is extremely important aspect to ensure tissue physiology and normal tissue function. And thinking that brain would give up on that really makes no sense. So it just pushed it, probably throughout evolution, just pushed it out into its borders, and that's what it's happening. But there is a but. Because if molecules from CNS are presented to immune system on professional APCs and are draining to lymph nodes via lymphatic routes, then what makes CNS immune privileged organ? Why then we don't get strong immune response when we get tumors in the brain? And actually this exact question Akiko Iwasaki asked herself, and what she showed 2 years ago now in this paper, is that if she actually takes tumors, puts them in the brain, but now activates and enhances the function of meningeal lymphatics with VEGF-C, she actually can enhance T-cell response against tumors, and in combination with immunotherapy, can really actually very nicely remove tumors completely from the brain. And there was a similar paper that came again weeks after hers from Jiankai Luo's lab at Peking University in China, and he showed very similar, his lab showed very similar findings, and they also show that if you ablate or if you kill those lymphatics, then tumors will grow even faster, which tells you two things, right? On one hand, you do have some immune response against those tumors, a base level, but probably for some reason, these lymphatics are limiting it and not working as efficiently as it could be. Now, that's one explanation, and it's lymphatic, and it's probably there. But maybe also there is something unique about the whole immune system around the brain. Maybe just the immune cells around the brain are not really poised to activate a strong immune response. In other words, to ask ourselves is maybe could it be that immune system and brain border is really very special? And 3 people in the lab really took upon this project. Andrea Kogura, who just recently graduated, he was my student in my second lab that I used to have in Germany, and he's now a postdoc with Michela Mattioli, just started his postdoc there. I already mentioned Tornike, a student here at WashU, and Justin, who is still officially a postdoc, but starting his lab very soon. So like I told you this, and so they were very interested in understanding this uniqueness of meningeal immunity. So as I told you, the meningeal immunity is quite diverse, lots of immune cells there, everything is fantastic. We, to be honest, were focusing on T cells, but then subsequently our attention drifted to other cells because we did this parabiosis of wild type and green mice. And after 60 days, we looked at T cells, and so our T cell story is hopefully will be out at some point soon. It's a very interesting, exciting story. I won't talk about it today, but as you can see, generally, even though there are some differences, which again I will not be mentioning today, but if you look at the number of GFE positive cells in this non-GFE mouse, it's about 40%, right, after 60 days. And then if you look at other tissues, there are some fluctuations, but overall there is not much difference. However, if you're looking into monocytes and neutrophils, you see a completely different story. So for example, monocytes are mixing quite okay in the periphery. You have about 30% of monocytes coming, green monocytes coming into non-green mouse. But if we're looking into the meninges, we are only about 10%. So why is the meninges only has third of the blood monocytes? I mean, that makes no sense, right? Because if it mixes, it should mix. All other tissue mix, liver mixes. So why doesn't spleen mixes? Why doesn't meninges mix? So we said, well, maybe a lifespan of monocyte in the meninges and of neutrophil is much longer. So it takes much longer for the cells to eliminate, and that's why the exchange with the blood is very, very slow. But that's not the case. Actually, the exchange rate, the lifespan of monocytes and neutrophils in the peripheral blood and in the meninges is very, very similar and very, very short. Okay, so that doesn't explain this. So what else could it be? Why is it? So maybe we have progenitor cells for monocytes and neutrophils in the meninges, but we couldn't find any progenitors. So how come you maintain a niche with the cells that normally come from the blood without really getting them from the blood, yet you maintain your niche? That made not much sense to us. But luckily, here in this case, we did know the literature very well. And so what we know is, of course, that the skull is a flat bone. And flat bones, as you all know, have a very high amount of bone marrow. And so skull is also a quite substantial bone marrow niche. And a few years ago, 3 independent groups showed that this bone marrow niche in the skull could actually come down directly into the dura using those unique channels, which they called, I think they call them skull channels, the dural channels. But the idea is that these channels are, it's an ossified structure. It's made of bone. Inside there is a vein, but the cells are crawling between the vein and this channel and kind of crawling down. So the first paper was from Matthias Narendorf's lab at Harvard. And what he showed that during stroke, neutrophils can take this route. That was the first demonstration that these channels could be functional. In this paper, Dorothy Sipkins from, I think, Hopkins, her lab showed that leukemia could also use this route to invade the meninges. And Alier Turkpub showed the beautiful structure of these vessels with his beautiful microscopy. So we thought, okay, maybe these vessels are not only used in pathology, but maybe they are also used in physiology. And the skull constantly supplies immune cells to its underlying borders, which is a cool idea, right? So you have this very important organ, and outside of it, you have immune system that kind of maintains this tissue. And so you have this unique reservoir of kind of private reservoir that constantly supplies you immune cells. It's like having a private generator. So if there is a power outage, you don't care because you have your electricity coming from your own private sources. Now, how do you prove it? So we did all the experiments which would suggest a local source, but how do you actually prove that cells are coming from the skull? So we decided to do a skull transplantation. Yes, sounds crazy, but it's doable, and you can do it, and we can happily share with you our know-how. So what we're doing is we're cutting this piece of skull from a normal wild-type mouse. We're taking a piece of green tissue from green skull, similar green skull from the green mouse, UVC GFP mouse, and plugging it in that area. So it's a little transplantation of calvaria bone. And we know that this area around the sinuses is very enriched in bone marrow. Now you can see the sinus, this area survives quite nicely. You can see after 30 days it's still there. And if we do slicing, we can see a bunch of immune cells in it. So the transplant survives. Now the question is, okay, can the cells really migrate into anywhere? Well, yes, because now if we're looking into the dura underneath the transplantation area, we can see a bunch of green cells. So we focused on monocytes, on DCs. We also showed B cells, but really the B cell story came from an accompanying paper from a colleague here, Marco Colonna, who showed really beautiful story about B cells coming from the same sources. And interestingly, there were now, I think, 2 or 3 papers already showing this B cell source being in the skull bone marrow. Let me show you quickly the channel. So this is how a channel looks like. You can see kind of protrudes into the skull, and then when the cell is coming, it is coming, as you can see, the green cell in between the blood vessel and this bonus structure. What we also showed, which hasn't been known before, is that not only the skull bone marrow, but also vertebra bone marrow have those channels, and the channels become very, very active with the cells during EAE. So if we do EAE in 2 parabionts, we're inducing EAE in both mice, what we will see is that, again, the mixture is about 25% in control mouse in the periphery, and less than 10% in the mouse. This is now looking into the spinal cord, into the CNS. So majority of the cells in EAE will be coming from local sources, but some will be coming from external sources. Now the question is, which one are the good cells? Well, so it could be the following, right? It could be the cells that are coming locally are the good guys, and the peripheral ones are the most aggressive. And our single cell data suggests that this is the case, because when we sequence the enriched for periphery versus enriched for local, we see this. So Alexander Meilner had this nice paper where he showed a phenotype of really aggressive monocytes. So we see the signature enriched in peripheral cells, but it could also be that maybe the cells are very similar and they just local sources are supplying as much as they can, and once they can't, then cells are coming from the periphery. So it could be good cells, bad cells, good cells, bad cells, or just equal type of cells. So we are looking into it to understand, because if there is really differential phenotype of cells and they are doing different effects, and maybe we can attenuate some of them and enhance the other ones, and maybe that's how we could maybe change the disease progression. Now, same results are happening with spinal cord. Again, majority of the cells in the injured spinal cord tissue will be coming from the local sources, and only few will be coming from the periphery. Optic nerve, even more dramatic results, differences. But of course, if you do a skin injury, then there is no difference, and there's no local source for monocytes. So they all come from the blood. Now, then actually we were doing something quite interesting. So this is a new unpublished data, and this was almost by serendipity, even though there was this idea of maybe CSF can mediate some signaling between the different tissues in the CNS. But really, when Jose, who is the MSTP student in the lab, and together with Leon Smythe, who is the postdoc in the lab and in collaboration with Justin, when they were looking at spinal cord injury, they actually looked at what's happening with the cells in the skull bone marrow. So you do spinal cord injury, we thought that maybe we can just use the skull bone marrow as a control from these mice. But what they realized actually is that every population of precursor cells, progenitor cells in the skull bone marrow will change after spinal cord injury. And we said, wait a second, how come you do spinal cord injury, it's so far away, and then it signals far enough to really affect the bone marrow in the skull? Okay, bone marrow in the vertebra, but how come bone marrow in the skull? So could it really be that, like what people were suggesting, Jose primarily, is that really CSF is flowing in between compartments and is being a signaling molecule? So they established this very cool flat whole mount of skull plus dura. So here we take a skull, we cut it out, dura is attached, we decalcify, and then we actually can whole mount it. And so this is labeled for the bone, for vasculature, and we inject this mouse before we killing it with a tracer. And you can see the tracer here in magenta. Now we can fly through different layers and observe where the tracer is. And again, we put the tracer in a CSF and we're taking the skull with the dura. So first of all, we see it, of course, within dural macrophages. Of course, it gets into the dura, but then it flies out. If we're flying up, okay, it should continue to fly. Why is it not flying? Okay. Okay, now we're flying, and now we start seeing it within the channels. Okay, now we see ovipositive channels as they're going inside the skull, and then eventually it actually gets in, we're getting into the skull bone marrow niches, and we can see that skull bone marrow macrophages are also being labeled. So the tracer actually goes through all the layers and reaches skull bone marrow. We can also see it in a live mouse. So this is a live mouse video, just to make sure that it's not some artifact of a dead mouse. But even in live mouse, and we're doing imaging of the niche, we can see tracer coming with it. This is maybe my most favorite experiment, because here they inject antibody just to label progenitors into the CSF, and what they can see is that from the CSF it will go into the dura, and then it will reach a skull bone marrow, and 100% of progenitors will be labeled by this method, and nothing in the tibia. This is exciting because it tells you that the signaling molecules from the brain can really reach the skull bone marrow niches and probably alter it. And we can also do it even if we put tracers inside the brain, they will come out and reach skull bone marrow niches. So the brain and its own reservoirs for immune cells are in constant interaction and collaboration. Now I must say that Matthias Narderov actually showed also a beautiful paper. This is now on bioRxiv. And I hopefully will accept it also very soon. So they're looking at a little bit different system with pathogens, but they're showing similar results. The CSF gets into the tissue. And again, Norwegian group from Ringston and Ida showed that this also happens in humans, and their papers have already been published out. So there is now a bunch of papers with ours out coming out, and actually Ali just published his on bioRxiv. There'll be like 4 or 5 papers really, all from different angles, showing how CSF can get into the tissue. And we also show that there is a functional consequence to it. So first of all, if we inject an inhibitor for SDF1 into the CSF, it will reach the dura, will interfere with SDF1 signaling there, a gradient there, and then we will have this rush of monocytes directly into the dura, because as you know, SDF1 keeps them there in the site. And indeed, you know, if we take a mouse which is spinal cord injured, and we then after 3 hours harvest its CSF, and then we inject it into a naive mouse, this is also sufficient to kind of call and rush these monocytes directly into the dura. So again, showing you that there is a constant interaction between these local producers of immune cells and the rest of the CNS. And this is what this figure summarizes, that you have signaling molecules from the brain are going all the way to skull bone marrow niches, and then these niches then provide immune cells into the dura, and under pathologies, such as I showed you EAE and I showed you in injury, the cells can actually go all the way down into the brain tissue. This is exciting, of course, because we're showing also now our paper that this skull bone marrow niche is a bit different than every other niche in the body, because probably just being constantly signaled by through the molecules in the brain and some probably unique interactions there. But just think of maybe a neurological disease that is happening, and it maybe send a message into the skull bone marrow niche, and it will change the niche maybe forever. And then these cells that are supposed to provide support and support to the tissue really won't be able to function potentially. So remember also the skull is a very interesting. Of course, it's a bone. It's very close to the surface. And these niches are actually potentially manipulatable. So you can actually reach the niche, you can remove the cells, and you can replace the cells. And so we're now working on how to alter this niche and how to change the cells and the phenotype of the cells within the niche. And then maybe when they're recalled into the damaged tissue, damaged brain, this will be just a natural path, right? Instead of jabbing cells into the brain tissue, maybe you can introduce them directly into the skull, and they will actually differentiate there and migrate their natural route. So I'll summarize here just very briefly that CNS is characterized by unique immune responses, but the mechanisms that underlie such uniqueness, they still need to be revisited. I still think we are not there yet. While there are no lymphatic vessels in the brain parenchyma, there is a lymphatic drainage from CNS via meningeal lymphatic vessels. There is also a bona fide immune surveillance of the brain, which happens from within its borders. Dural sinuses serve as immune interface. CNS antigens are presented there to patrolling T cells. And the skull and vertebral bone marrow niches are immune cell reservoir for meninges and likely for parenchyma under pathological conditions. And finally, I showed you that CNS kind of controls its bordering bone marrow niches via soluble molecules delivered from the brain within the CNS. So of course, our love to lymphatic vessels is still strong. We love the lymphatic vessels very, very much. We think that this is a very interesting aspect of immune uniqueness of the brain. But we are now also really excited about the skull and its immune niches within the skull, and understanding how these unique niches may also be playing part in brain immune privilege is kind of probably the next chapter that we are going to look into, and it's very, very exciting. So this is probably the most important slide, is to thank all the, of course, funding sources and many collaborators. Unfortunately, I didn't have time to present any of our Alzheimer's work today, that we collaborate with Dave Holtzman very closely, and many other interesting stories that we have collaboration with Marco and with Gwen and many other people. And also outside of WashU, also some exciting collaborations. Actually, I should switch. Kodi Ravishandran just moved to WashU, so I should move him here. And then, well, maybe arguably this is the most important slide. So this is the lab. We had a couple of weeks in October where we were allowed to go out and take a picture without mask and show our more updated picture of the lab. But I think I showed each person individually who did what as I was presenting. Maybe I just want to feature Igor, who is our animal surgeon and does really all these exciting surgeries and helps everybody in the lab. So I'll stop here. I'll just say that we always welcome curious and talented postdocs to join our lab. So never be hesitant to just reach out by email, and we can talk about your interest. So I'll stop here. Thank you very much.
Thank you very much, Jony. Thanks for this this tour de force, really fascinating, really work. So there is no question. So I just wanted to share with the people listening to your talk that if they want to ask you a question, this will be done via Twitter, where you will first search for the account, the Global IMMUNOTALKs, and then find the tweets that say ask question for Dr. Jony Kipnis. And after reply to that tweet with your question, and of course mention Global IMMUNOTALK. Otherwise, yeah, you can contact Jony at his Twitter address at Jony Kipnis. Thanks a lot, Jony, and see you soon. Bye bye.
Bye bye. Thank you.
Thank you very much.